Look at the contraction of a muscle, specifically a skeletal muscle here. If you have a muscle, skeletal muscles are attached to bones. They move bones at joints, and an entire muscle is made up of a bundle of muscle fibers, and those are the cells. They're long muscle cells bundled in fascicles, and then the fascicles are bundled into the muscle. There's one nerve that innervates an entire muscle. So one nerve from the spinal cord will innervate that muscle. However, at the end of the axon, at the end of the nerve cell, it will branch and branch and branch into axon terminals, and these axon terminals will touch each muscle fiber. There will be a muscle fiber innervated by some branch of the nerve that's innervating the entire muscle. So at the end of the axon, you have the place where that the axon touches the muscle fiber or the muscle cell, and it happens at a place called the neuromuscular junction. It just means where they touch. And they are touching right above the sarcolemma of the muscle cell. And the sarcolemma is the membrane of the muscle cell. And they actually do not touch exactly. There's going to be a space between them. And when the action potential or the nerve impulse rushes down the axon to touch to the muscle, you're going to set up essentially a new nerve impulse in the muscle cell itself, and that's what we're going to look at today. So this is the end of the axon. This yellow structure is the terminal bulb of the axon, and it's located directly above the sarcolemma. That's the bottom part, the muscle fiber itself, and there is a space here, and that space is fluid filled. So you're going to have not just water above, on both sides of the cell membrane. You're also going to have ions that have been pumped across these membranes. So when the action potential rushes down the axon, and I think that's indicated by the pink arrows here, what's actually happening is you've got ions being dropped out of the cell to the outside, and then new ions being brought into the cell, and there's a sequence of events that goes down that membrane. What's happening is it's actually changing the voltage of that membrane. So the positiveness or the negativeness of the inside and outside is changing, and that's picked up by a trigger in a calcium channel right at the end of this terminal bulb. So this channel will open when there's a certain voltage regulated in the membrane. Calcium will rush into the terminal bulb, and when that happens, it's going to promote a fusion with the synaptic vesicles. These vesicles are bubbles holding a neurotransmitter called acetylcholine, and that neurotransmitter allows a nerve impulse to go to the end of the axon and not stop, but actually be picked up by the muscle cell, and then essentially you're going to get an action potential in the muscle cell that will cause the muscle contraction. So this acetylcholine is a chemical that when it fuses with the membrane and dumps its contents into the synaptic cleft, it will diffuse across the synaptic cleft. Now, we saw that the calcium channels were voltage regulated, but the acetylcholine channels, or I'm sorry, the channels on the muscle fiber itself are triggered by acetylcholine, which is a molecule. And so that's called a ligand-regulated channel, and this ligand in this case is the acetylcholine. So when acetylcholine acts like a key, it opens up the channel inside the muscle fiber itself. So this channel, the green dots represent the acetylcholine that's touched that receptor site. This channel opens, and a couple ions will rush in and out. Sodium ions, which are positive, is going to be packed on the outside. It's going to be piled up on the outside. Potassium ions, which are positive, also are piled up on the inside. When this channel opens up and both of them are able to flow through freely, then sodium rushes into the cell and potassium rushes out of the cell. What that's doing is it's changing the positive and negative quality of the inside of the cell and the outside of the cell. It's basically messing with the voltage. And this voltage is going to be picked up by new voltage regulated gates in just a bit. So just to tell you that once this key is in the lock and the gate is open, in order for a new muscle response to happen, or a new muscle contraction to happen, these have to be broken down. You have to take the key out of the lock so that the lock will close so that this can happen a second time. So there is an enzyme, and the enzyme is called acetylcholinesterase, and acetylcholinesterase will break down the acetylcholine, and then it will diffuse back across the synaptic cleft and be taken up again by the axon terminal and restored into the synaptic vesicles as acetylcholine. So it breaks down and then is reintegrated back up into the axon and put back together into acetylcholine so that the nerve impulse can work again. So these have to be repeatable over and over again. The proteins have to move back and forth. The gates have to move back and forth. These chemicals that are acting as neurotransmitters need to be back where they belong so a new nerve impulse can happen, so a new muscle contraction can happen. So this is again under the synaptic cleft, and you'll see that the acetylcholine is diffusing across the synaptic cleft and has touched these ligand gates, which open up these sodium-potassium gates, which changes the polarity. So it changes the amount of positive and negative quality inside the membrane. Now normally a membrane is negative inside and slightly positive outside. That's normal. So the voltage on the inside of a normal resting cell, a muscle cell, is about 70 millivolts. So that's how much positive compared to negative it is. So it's slightly negative on the inside. When these sodium and potassium gates open up, more sodium is rushing into the cell than potassium is rushing out. And when that happens, the inside becomes less negative than it was, slightly less negative. So when the membrane becomes about negative 55 millivolts, a sodium channel will open up. And that's a voltage regulated channel waiting for that voltage, and as soon as it's reaching that voltage, it opens up. Now remember, sodium is being pumped to the outside of the cell, and that sodium will rush into the cell, making it more positive still. So it was at negative 70, then it comes up to about negative 55, and then it keeps rushing up. So let me show you quickly. This is negative 70 is normal. And then when the sodiums and potassiums change places, it slightly raises the charge inside the cell to negative 55, which triggers the sodium channels to open. When the sodium channels open, sodium ions flood in, which makes it even more positive. So that's a positive feedback loop. And when it gets up to about 30, the sodium channels, which are voltage regulated, will close, and the potassium channels were open. So I'll go back here. So what's happening is after the sodium will close at 30 millivolts, this is called repolarization, potassium will rush back out of the cell, and because the sodium channel is open, and that's going to make it more negative again. These positive potassium ions are going to flood out, which reestablishes that negative 70 millivoltage inside the cell. So what's happening, it's a very temporary place. It is positive only at this very, very small location, and then it flips back to negative. But as it flipped positive, what it does is it triggers the next set of sodium gates, which will open, make it slightly positive inside, which will open the potassium, which will bring it back to negative. So it's a sequence. One happens, and then the next happens, and then the next happens, and it goes down, propagates down this membrane, basically opening channels back and forth. This is exactly what happened in the nerve cell itself. The nerve cell had potassium and sodium that will flip, and change the polarity at that very localized spot, and then it triggers the next gate to open, and then it flips the polarity, and the nerve impulse, that electricity rushing down the nerve, that's what's actually happening. It is chemicals flipping back and forth, which are changing the amount of positive and negative quality of that membrane, and that's what electricity is. Electricity is negativeness or positiveness at a certain location. And so your nerve is so electricity could run down your nerve, and chemical, so this electricity is in the form of ions flipping places. So what's going to happen there at the terminal bulb of the axon is that a depolarization wave is propagated in the muscle. And so now the same thing that happened in the nerve, essentially a nerve impulse, is rushing along the sarcolemma of the muscle. That sarcolemma is the membrane of the muscle. And as it flips back and forth, you get this red arrow here, which is heading down the muscle membrane. Now, you're going to get to a hole. There is a hole in the ground all over the place, and this hole will essentially go into the muscle fiber itself. So think of it as a tunnel that'll go from the membrane that goes into the cell, and that membrane or that tunnel is called the T-tubule, and the T-tubule will go into the muscle fiber itself. Now the T-tubule essentially is the sarcolemma that goes down into the cell. So really it's part of the membrane of the cell, but it's internal. So think of the wall that surrounds the entire muscle as going deep into the muscle cell. And so this impulse that's rushing along the sarcolemma of the cell rushes down into the cell. There are gates in these T-tubules that are connected to the sarcoplasmic reticulum. If you remember biology, the endoplasmic reticulum is essentially a network, like a river system or a tunnel system inside the cell, where all kinds of things happen kind of in a quicker manner. Well, there is a swollen area on both sides of this T-tubule, and this swollen area is called the cisternae, and it just means cistern, like where you store water. And this terminal cisternae is essentially a swollen area of the sarcoplasmic reticulum filled with calcium, and the calcium are in ions, positive 2 ions. When the action potential rushes down the T-tubule, it triggers these gates to open, and these calcium ions flood out of these storage locations and into the entire internal muscle cell. So the whole inside of the muscle, the cytoplasm of the muscle, is flooded with calcium ions. So what's happening with the calcium is that there is a receptor site on a chemical, on a protein called troponin. And when calcium touches the troponin, it changes shape. The troponin is connected to a band or a ribbon of a different chemical or different protein called tropomyosin. And tropomyosin is acting as a cover of the myosin binding sites. So you had the thick and the thin filaments in the sarcomere. So now we're down onto the really small, essentially chemical level of the inside of the muscle cell. And you have a myosin fiber, which is a thick fiber, that have these myosin heads embedded in this fiber. And then you have a thin fiber called actin. The actin has a place on each molecule that the myosin head can easily connect to, easily without expenditure of energy. But they're covered. The tropomyosin is covering these binding sites. So normally a muscle does not contract. A muscle only contracts when you tell it to contract. When your brain sends a signal down your nerve to your muscle, that's what's exactly happening. that goes all the way down in, crosses this synaptic cleft, propagates a new action potential on the sarcolemma of the muscle fiber, rushes down the T-tubules, opens the calcium in the terminal cisternae, and the calcium rushes into the sarcomere. These calcium touch the troponin, just like a key, and the troponin changes shape and pulls the tropomyosin off the actin binding sites. When that happens, then the myosin head automatically attaches. It automatically attaches to these active sites that are exposed, and you've formed a myosin cross bridge. So the myosin and the actin is now touching. I think of it almost like Velcro, where they're actually bound to each other. So what's going to happen, this is going to happen repeatedly, because it's almost like these arms are grabbing, pulling, and as they're pulling, these blue actin filaments are going to be pulled in, and this sarcomere, which is this contractible unit of the myofibril inside the muscle fiber, is going to be pulled in and contracted. So what will happen is you have got a energy molecule called adenosine triphosphate, ATP, that has bound to the myosin head. When it bound to the myosin head, it changed shape. The myosin head changed shape and cocked like a gun so that it was ready to pop onto this actin filament. That ATP is broken apart into adenosine diphosphate plus that additional phosphate. So when that phosphate was broken off this molecule, the energy from that bond was able to change the shape of the myosin head. Well, the myosin head now attaches normally, attaches for free, but it's going to get rid of this ATP and phosphate and change shape again. So in the second step, it pops off the adenosine diphosphate and the phosphate, and it changes back to its normal shape. Well, its normal shape is constricted, and as it does it, it's attached to the actin, and it pulls it. And so that's a free action. It pulls the actin, and that's what's actually contracting teeny little bit of the actin molecule in towards the midline of that sarcomere. Now, if a new ATP then binds to it, so ATP is like money. I need another dollar to spend. The ATP binds to it, it rechanges shape. And as it rechanges shape, it detaches, the cross bridge, it detaches away from the actin, and then as it breaks it apart, it's going to recock. So that breaking of the ATP changes its shape, and this is the same thing. We do it again and again and again. The ATP has changed shape, or it has changed the shape of the myosin head when it's broken apart. When that last phosphate group is broken off the ATP, making ADP, it will now be able to pop right onto this binding site, and then it just repeats. So it just attaches, and then it breaks apart, pulls it and contracts the muscle, then a new ATP binds, and then it breaks it apart, then it's ready to attach again, and it reattaches again and again, and it just repeats this again and again. So what's happening is the actin is being pulled in towards the middle, and so the red here is the myosin, that's the thick filament. The blue is the actin, that's the thin filament. And since the thin is being pulled in towards the midline, that M line, you're going to get a contraction of this sarcomere. Now, this is a sarcomere here, and then the next sarcomere is next door to it, and you can see that there's multitudes of sarcomeres. They are stacked up and down, round and round, in and out, side by side, and then the more of these that contract, the stronger that muscle is contracting. The more you could lift, for instance, or whatever, or act as leverage. So this is a contraction. This is a very detailed lecture, and I want you to learn it, and I will help you in any way I can. I wish you good studying.